morning. Yeah. I'm eating these little tiny tomatoes, so I'm trying to check that I don't have any in my teeth, but they're pretty good. Yeah. Anyway, hi, this is Dr. Ben. Let's go. So, our first vaccine related paper of the day is it's called Single Dose Replicating RNA Vaccine. We've hit a snag already in the title, but we'll get to that. Induces neutralizing antibodies against SARS-CoV-2 in non-human primates. Yeah, all the rest of the title is true. The first word or hyphenated word. That's where the problem is. So this is an interesting concept, and I've heard this group talk about this sort of thing before. So this is coming through a guy called Ari Helenius, I believe. Somewhere on there, I think. And Heinz Feldman. Yes, these are big names that run massive laboratories. Anyway, the idea is that you can take a different virus. So the idea is that a replicating virus, a virus that's growing inside you, is going to tweak the immune system in different ways that a killed and activated virus will not. And so the neat thing is that these tweaks, these are things that are going to go on inside the cell that's actually infected, and it'll start to realize that things are wrong. Some wrong things are happening. We're copying RNA outside the nucleus, and we shouldn't do either of those things. And then it will start to make a response, and that response actually ends up contributing way down the road, like 10 steps later, to actual development of an immune response, and like how strong the development of the immune response is. So it's a useful thing, but it's also a really dumb primitive system that can be triggered in a number of ways. And so smart scientists are trying to find out a way to make the cell think it's infected and make the cell make a protein from SARS coronavirus 2. This is the spike protein. And so at the same time, it'll say, whoa, I'm infected. Here's SARS coronavirus 2. Please make an immune response against this. And it's a pretty good idea. So what they've done is they've taken the VEEV, Venezuelan equine encephalitis virus, which is an alpha virus, which I don't know why. Yeah, if you care about these things, it's another positive strand RNA virus, but it's pretty different from coronaviruses. But they have certain things in common. There are a couple enzymes that kind of do the same thing. There's nothing like that would cross-react between the two. They're not close enough that you could make an antibody to one that does anything to the other. But they're both RNA viruses, and they both work in the same enough way that theoretically they're going to set off similar pathways inside the cell. And so that could be good, right? Yeah, it could be good. And so they've got this replicon, which is just the proteins that are needed to copy the RNA that they're embedded on, and that's all they do. It's just like a little self-copying thing, but it is missing the other part that makes a virus, which is some way to spread. Usually there are one or more proteins that make some kind of box or shell around the instructions so it can go to another cell and then start another infection. That's the total idea of a virus. They're very simple, kind of stupid little things. Anyway, so what they've done is they've taken just the copying part from VEEV, and they've put on the spike gene from SARS coronavirus 2, and they've set it up so that as this is copying itself, it's also going to make copies of the spike. It's just going to run off a couple extra little copies of that, which will then get turned into protein. And so, bam, you have what they call a replicon, because it replicates, but it doesn't infect. And it's able to make the SARS coronavirus spike. And so you say, well, that's cool, but how do you actually get that into a cell? And they do it the same way as the Moderna vaccine. So it's in a little lipid blob of some sort, and I'm not sure they specify, or maybe I just didn't read that part closely enough. But whatever, there are ways with lipids that you can get a piece of RNA into a cell. So yeah, we'll take that as given. All right. Well, Dr. Ben, what are your problems with this paper? Here are my problems. So at the start, the paper actually looks really good. Their replicon works. They make antibodies. They get decent antibodies in mice, which is great. But remember, it's not about how many antibodies you make. It's in part about how many neutralizing antibodies you make. That's not the whole thing. You really want to make, reactive T cells and a whole range of things. Yeah, helper T cells, killer T cells, and antibodies, and you want, the proper regulatory cell environment. You want to make sure you're turning off some of the cells that are going to cause a lot of damage if they overreact. You need the total package. Yeah. But the easiest thing to measure is neutralizing antibody, or is antibody, yeah. And antibody is not very useful, but neutralizing antibody is somewhat useful. So it's a good correlate. It means if you've got that, probably, maybe, but we'll find out, but probably you have other parts of the immune system that are also there and able to recognize the virus. This is the idea. Yeah. And so we scroll down to, no, not figure 4, I think it's figure 5. There we go. Yeah, and they call it the lion, which stands for, I don't know, it's terrible, but the L is, I think, lipid. The lion rep RNA CoV2S. Wow. Yeah. And so with one dose, so they say single dose up there in the title, they get a neutralizing antibody titer of between about 30 and about 50. Yeah. And in people who have recovered and are probably going to be protected against the second one, we see neutralizing antibody titers of 500 and up. And so if you're averaging, a tenth of that, then you're probably not protecting. Yeah, you may be helping a little bit, but you're probably not actually giving protection at this level. So the single dose does not seem to be super duper immunogenic. Problem is, in the monkey models, you don't need quite as strong an immune response to actually be able to knock out the virus and clear it. That is that's the issue here, really. So then they gave the virus or the vaccine twice, which is why, again, I say single dose. Single dose didn't work very well. Double dose worked better. With the double dose at 2 different dosage levels, they actually end up with around neutralizing antibody titer of 200, which is you're about halfway to what would be considered reasonable. Like a reasonable lower limit for a vaccine. So this is better. And I don't know if you gave this a third time, if that would finally get you enough of an immune response that you could be able to actually fight this off. So they do some tests in here, and it seems to give a good immune response in the monkeys, which I think they did in pigtailed macaques, which I'm just imagining little pigtails on the side. I don't know what a pigtailed macaque looks like, but good for them, good for the macaques. Probably bad for the macaques, but well, whatever. Yeah, that's science. So it's not terrible. It's not the answer yet, and I'm not sure how they can tweak it. They did notice that if they gave more of the replicon, they got more of a response. So they may be able to crank up the dose, but at a certain point, when you crank up the dose too high, then you start to get side effects from all the lipids running around, all the RNA running around. These set off kind of non-specific pathways, potentially, that could anger your immune system or other cells, or just damage some of the filter organs. Maybe, I don't know. And you don't want to just be making this, because I think a lot of it is going to end up being expressed actually in those filter organs, and you need them to do a second job as well, which is keeping your blood fairly clean of metabolites so that you don't die. But this is positive. There may be ways that they can modify this, get it to express in a different tissue a little bit better, or change the dosage. So anyway, it's another idea. It's been tested out in primates, and so that's a whole lot of money behind it. And it's coming from some people that are pretty smart and have done some good work in the past. So I don't know. That doesn't really mean anything for the results, but makes you say, well, at least these people kind of know what they're doing. And there you go. That's your first Dr. Ben vaccine update.